Shall I start?
Sorry, Ulaş will introduce you in few sentences.
Okay.
Or you can introduce yourself as you like.
No. Okay, well, I can introduce myself.
Okay, so please go ahead and introduce yourself.
Okay, well, thank you for having invited me to give this webinar. My name is Peter Lenting. I'm a director of research working for the French National Institute for Health and Medical Research. We are located at the southern part of Paris, in the hospital on the photo that is here at the bottom. Wait, I will try to point my the laser pointer. Yeah, so here in the southern part of Paris, in this hospital, which is one of the older hospitals in Paris, actually. And so what I want to do today is to talk about one of my favorite proteins. I have a few. Factor VIII and VWF are the two most important ones. And today I will talk about Willebrand factor, about its role in and beyond hemostasis. It will be a long story because I will talk about different things. In the first part of my presentation, I will talk about VWF structure and function. And there I will focus mostly on the biosynthesis and secretion of the molecule and talk a tiny bit about its clearance. And then presenting a bit about the main function of VWF in primary hemostasis. I will not go very deep. I will just touch on a few things which I think are essential to understand how VWF is working. And I will give one example about the role of VWF that is beyond hemostasis, and more particular on smooth muscle cell proliferation. And in the last part of my presentation, I don't think I can afford to talk a bit about VWD, Willebrand disease, and a bit more about what is the actual burden of VWD, and give a few examples, and actually one example in detail about new treatment options for VWD. So let's start to go ahead with biosynthesis and secretion and clearance of VWF. So VWF is a particular protein in that it's produced in different tissues. The main source of VWF, we can find it in endothelial cells. You can see here on this photo where the endothelial cells from small arteries are staining very intensely for VWF. And when you go into more detail, you can see that within the endothelial cells, VWF is located in small organelles, which we call the Willebrand bodies. From the endothelial cells, VWF is released not only into the circulation, but also into the subendothelial matrix, and I will come back to that a bit later. Another source of VWF is in the megakaryocytes, in the bone marrow. You can see some megakaryocytes that are stained very brown, very intense for VWF. And via the formation of proplatelets, those little things that are here when you start to stimulate megakaryocytes, they end up in the platelets, and platelets stain positive for VWF. So when you finally look at the different compartments where you can find VWF, there are a few. As I say, you can find VWF in plasma as a free protein, but also in platelets, in the alpha granules. You can find it in the subendothelial compartment, and also within the endothelial cells in the Willebrand body. So we have 4 different sources where we can find VWF. So how is VWF synthesized? And I'll focus here on what's happening in endothelial cells. So VWF is a molecule that contains a number of different domains. It's a very defined structure with the architecture of the different domains. They start with D1, D2, the D prime D3, it's the D domains, and we also have a D4 domain. Then it's followed by 3 A domains, and at the C-terminal part we have 6 repetitions of what we call C domains, And finally, there is a cysteine knot domain that keeps the structure in a dimeric form. So when this large molecule, which is about 2,300 amino acids long, so it's very long molecule as a monomer, when this dimer is being formed in the plasmatic reticulum, it starts to form some kind of bouquet structure. And that's only happening at low pH, about pH 6. And when this is happening inside the cell, actually these bouquets, they start to form a helical structure with each of these arms presenting one of these dimers. And when this helical structure is being formed, and then we are already arrived in the Golgi, then these long helical structures are actually packaging into what we call mini stacks. And these mini stacks then start to form the viable pallada bodies. So when you take out VWF from these viable pallada bodies, then they start to form these very long multimers. So how are these multimers are built up? Well, we start very simple with the dimer. You can find these dimers in the circulation and in the endothelial cells. And that is where a dimer consists of a CK domain, two CK domains, the cysteine knots that are being linked together, and without the propeptide being there anymore. So I go back here. So I mentioned here the D1, D2 domain that are at the N-terminal part of the Willebrand factor, and this D1, D2, which is also known as the propeptide. And this propeptide is actually cleaved off during synthesis in the viable pallada bodies. But this propeptide is very important because it will help to multimerize the molecule. The propeptide helps to a second linkage between those dimers, where the D prime, D3 portions are being linked together. Well, if you continue to do that, then on every step you will add an extra dimer, so the molecule starts to grow into this multimer. So you start with a dimer, which becomes a tetramer, an hexamer, an octamer, and decamer. You can actually go very large until you have more than 50 of those dimeric subunits being linked together. So you have more than 50 mers with a molecular weight that is more than 20 million Daltons. So it's actually in plasma, it's the largest molecule that that is circulating. So once it is located in these viable pallada bodies and being produced in the endothelial cells, it will be secreted either to the plasma compartment, and some small portions will also being secreted to the subendothelial compartment. Well, without going into the very details on how these different secretion pathways occurs, but you can actually distinguish three distinct ways in which VWF is being secreted from the endothelial cells. The easiest way is when there are just some small vesicles going directly from the Golgi to the plasma membrane of the cell, and then these small multimers are being released without being forced to go through a viable pallada body formation. We also have the occasion where a single viable pallada body is actually fusing with the plasma membrane of the cell, and then the content is being released. And that is also secreting multimers that are a bit of intermediate size molecular weight multimers. When this is happening, there is also secretion of other molecules that are being stored in the viable pallada bodies. And finally, when the cells are stimulated, endothelial cells are stimulated, what is happening is that several viable pallada bodies, they can fuse into one, what we call secretory pod, and then the content of the secretory pod is being released. And it's actually in this case when those very large multimers are assembling at the endothelial surface, and they are forming what we call strings. And actually these strings you can actually see under a microscope. This is one of the examples that we have done a few years ago in our lab. So here on top what you see is those endothelial cells which were then exposed to flow that contained platelets. And what you can see is that those platelets, they start to form those very long strings, with each dot being one single platelet. And when you then start to stain these strings for Willebrand factor with monoclonal antibodies, you can see that these strings, they can get very large. They can be even several millimeters long. So with a microscope, it's very easy to see them. And when you look into more detail, what you also can see is that these strings are actually composed of multiple Willebrand factor molecules. So you can see here some bifurcation in the string. That means that there are several multimers are actually sticking together to make up these bundles. So under the influence of flow, Willebrand factor unfolds into these elongated bundles of multimers that are able to function as an adhesive surface for platelets. And via what we call self-association, there are several bundles that can form, that consist of multiple VWF multimers. So once VWF is in the circulation, of course, it also needs to be cleared again. It needs to be removed because sometimes it's getting too old, it will lose its function. And by now we know that there are a number of tissues and a number of cells that are important for the clearance of VWF. In particular, macrophages and sinusoidal endothelial cells are very good catchers of the Willebrand factor molecule. And we also know that hepatocytes under some conditions can contribute to the clearance of VWF. Well, since we know that sinusoidal endothelial cells can be found in different tissues and also macrophages can be found in different tissues, it's not surprising that different organs are able to contribute to the clearance of VWF. And in particular, the liver and the spleen are relatively important for the clearance of VWF via the macrophages and the sinusoidal endothelial cells. So what kind of clearance do we have? Of course, there is the basal clearance, where just normal VWF is being removed from the circulation, and it's there where macrophages and sinusoidal endothelial cells are important. But we also know that there is some kind of quality control clearance. Under some conditions, VWF may not be good, or maybe have a modified conformation, or is perturbed in some other way. And that is where the quality control clearance starts to play a role. And there are two different ways in which that can happen. One is when VWF, which contains glycans that are normally very well sialylated, in some conditions, the sialylations are very poor, so your molecule becomes hyposialylated. And that is what we find in some conditions of Willebrand disease, in particular Willebrand disease type 1C, where there is accelerated clearance, but also, for instance, when there is increased bacterial infection, where these bacteria start to secrete neuraminidase, sialidase molecules that take off the sialyl glycans of VWF, and in that case, your VWF will be cleared more rapidly.
Sorry, Peter, can we ask questions, or shall we wait until you end?
Whatever you want. If you want to ask questions right now, that's fine for me.
Yeah, I have a question. So for this quality control clearance, it is for example, for you wrote VWD type 1C. If the Willebrand factor has mutations or it didn't fold properly, right? It occurs this type of Willebrand factor.
Yeah. So what we know, and that it's a lot of work by James O'Donnell from Ireland, who has done that, who has been looking in that, is what we see is that, and we did some ourselves too, about 15 years ago, is that when you look at the rate of sialylation of molecules that contain a mutation, for one reason or another, they are less well sialylated. We don't know exactly why, but they are less sialylated, and that has a consequence that those molecules are cleared more rapidly, and that can contribute to the low VWF levels in these patients. James O'Donnell from Ireland, who has done that, another quality control we have seen is that one of the receptors for VWF, LRP1, recognized 15 years ago, only when it's in an active conformation. Great acetylation of molecules that contain a mutation, and in those for one reason or another, they are less well cleared more rapidly because it starts to interact more. They are less cleared easily with this LRP1 receptor. So in terms of clearance, we have two sorts. We have the basal clearance, and there are a number of receptors that have been identified now. We also have some quality control receptors like the MGL1 and LRP1 that contribute to the removal of VWF that is not in the proper conformation. The role of glycans in VWF clearance it's also in the apparent from the clearance of blood group O and blood group non-O. So if we look at the glycans that are present on the VWF molecule, there are quite some. There are a number of O glycans that are mostly clustered around A1 domain, and we have some N-linked glycans that are a bit everywhere on the molecule. And each of these glycans, they are capable to contain ABO blood group determinants. And what we know is that clearance of VWF is more rapidly in individuals with blood group O compared to individuals with blood group non-O. There is a few hours of difference in the clearance. Now, is it the glycans on VWF that cause that difference in clearance, or is it the blood group status of the individual that makes that difference? That's not completely clear, but the latest evidence tends to go to the status of the individual rather than that of the glycans on VWF when it comes to the blood group determinants. So if I summarize this first part, so VWF is produced in megakaryocytes and in vascular endothelial cells. We can find VWF in storage organelles, in the alpha granules in platelets, and in the viable pallada bodies in endothelial cells. But we can also find it in the subendothelial matrix and in plasma. We know that there are different modes of secretion that are existing. We have the basal secretion, where it's usually the low and intermediate molecular weight sized multimers that are being secreted, versus regulated secretion, where VWF is where endothelial cells are being stimulated, and there we get the very high multimers that are being secreted. When we then go to clearance, it's a rather complex phenomenon in which many receptors are being involved, and in which we also can distinguish basal clearance and quality control clearance. And especially glycosylation plays an important role in the regulation of VWF. So what about function of VWF? Well, most of you probably know that VWF plays an important role in the adhesion of platelets, in the recruitment of platelets to thrombus formation, and it's doing that in a rather complicated way. A second role of VWF is to function as a chaperone for factor VIII, but that is a topic I will not go into today. So I will focus a bit on the platelet binding sites for VWF. So when we have here the molecule again with the domain structure with the different domains, we can identify a number of binding sites that are important for this platelet interaction, platelet recruitment function. So we have the binding of the platelet receptor GP1B alpha in the A1 domain. We have a number of collagen binding sites in the A1 and in the A3 domain. We have a binding site for the other platelet receptor, the integrin alpha 2b beta 3 in the C4 domain with an RGD sequence. And what I've also started discussing is a site of proteolysis for ADAMTS13 in the A2 domain. So by now, most of you probably know how VWF is actually working in the recruitment of platelets. So what's happening very briefly, so we have globular VWF, that is VWF that is really some kind of a little balloon that is circling around in plasma. When there is a vessel injury, your VWF will start to bind to collagen via the binding sites in the A3 and the A1 domain, and by that it will start to elongate. It will take another conformation. And when it is in its elongated conformation, we call it then its active VWF, that allows platelets to arrive and start rolling and finally adhering to VWF on this collagen surface. Three other receptors on platelets other than GP1B, like GP6 and alpha-2 beta-1, the platelets will also start to interact with the collagen, and the platelets will become activated, and that will lead to the recruitment of additional platelets. So in this way, the platelet starts to aggregate and starts to form a large thrombi. What is important is when your thrombus starts to grow, the shear stress at the edge of the thrombus will become stronger and stronger. And it's especially there where VWF will start to play a very important role, because only VWF under these high shear stress conditions is able to recruit additional platelets, and that will finally lead to occlusion of the vessel. So without VWF, you never get occlusion of the vessel.
And this shear stress is higher during the cut of the vessel, or when the shear stress gets higher?
Well, the moment that your thrombus start to grow, then the space between the lumen and the thrombus starts to get smaller, but the pressure of the blood, especially in the arteries, is remaining constant. So that means that there is very high pressure, a very high shear at that small portion of the vessel that is still open, and that will lead to disturbed blood flow, elongation of flow, etc. And without VWF, platelets will not be able to adhere anymore. So then the vessel will stay open. But if you have VWF, then the still platelets can be recruited, and then that will lead to occlusion of the vessel.
Okay, so the occlusion also enhances the shear.
Yeah, exactly. Well, one of the important things is that VWF is always there, and normally you should have a way in which you regulate the manner by which VWF is actually interacting with platelets, because they are both in the circulation and they only should start to interact at the moment where it's good, but it should also stop at some point. For that, there is some regulation in place. For instance, one way to regulate VWF function is to regulate its multimer size. We have already seen before, when VWF is being secreted from the endothelial cells, it can reach very long strings. It can assemble into large bundles that are spontaneously interacting with platelets. And actually, those long VWF multimers, the ultra-large multimers, they are prothrombotic. And in order to prevent that those platelets and VWF interact spontaneously in the circulation, VWF is actually cut in smaller pieces. The ultra-large multimers are being removed. And that is done by an enzyme which is called ADAMTS13. And indeed, if you compare the multimers that are present in endothelial cells and what you see in plasma, you can see that those ultra-large multimers are no longer there. This is a very important step, because if you do not have any ADAMTS13, you get all those platelet VWF aggregates, and they will start to occlude the microvessels, and that is a condition which is known as thrombotic thrombocytopenic purpura, which is basically lethal when it's untreated. But there are also conditions in which ADAMTS13 is actually its job doing too good And one example is when you have patients with type 2A, where there is a mutation that accelerates cleavage of ADAMTS13, and then you actually start to lose the higher multimers as well. And the problem there is that your VWF does not function good anymore. It will not have the hemostatic power to recruit all your platelets. So regulating VWF multimer size is very important. So it should not be too large and it should not be too short. You should just have the appropriate range of multimers that we normally find in plasma. Sorry. So how is this regulated? As I mentioned, in plasma, VWF circulates as a balloon in a globular structure, where it is unable to bind to platelets, but where it's also resistant against proteolysis. When there is increased shear stress, your VWF starts to unfold into a long elongated molecule, where you not only have platelet binding, but where you also can get the proteolysis. So at what type of conditions can actually ADAMTS13 proteolyze VWF? Well, the first thing, the most known part is that at the surface of the endothelial cells, where those large multimers are being secreted, well, when you have ADAMTS13 at that point, it will actually start very efficiently to cleave your multimers into smaller portions, and that is where you get rid of the ultra-high multimers. But we also know that if you infuse recombinant VWF in plasma in a patient and it starts to circulate, already after 10-15 minutes you can see that the VWF is being cleaved by ADAMTS13. And actually it's doing that at, for instance, at bifurcations of vessels where there is some disturbed blood flow, and that is very efficient. And finally, and that is also an important portion, is when you have platelets starting to aggregate into the thrombus, then VWF is being part of that. And in order to prevent that the thrombus is growing too fast or too big, the VWF that is at the edge of those thrombi, they are also being cleaved by ADAMTS13. So that will limit the growth of the thrombus. So it's a natural way to prevent that the thrombus is starting to occlude the complete vessel. Well, sometimes that works and sometimes that doesn't. So we have VWF platelet interactions that are very strongly dependent on VWF multimer size. And the multimer size is mostly regulated by ADAMTS13. But in order for ADAMTS13 to cleave VWF, we need shear stress induced unfolding of VWF, and that can occur at the different positions, at the surface of endothelial cells, at bifurcations, or at during thrombus formation. And this latter part is important to prevent the release of ultra-large multimers in the circulation and to limit thrombus growth. So this is what I have presented here. Is for me it's a bit like textbook conditions of VWF, and if you want to learn more about VWF, I mean, this is the basic part that you would need to know to understand how VWF works and how what is its role in primary hemostasis. So now I want to shift the gears a bit and start to talk about some parts of VWF that are less known, less well understood also at the same time, and that its role is its role beyond hemostasis. So we now know that VWF is not only involved in hemostasis, but also in other physiological or pathophysiological processes, which is not a big surprise in a way, because VWF is such a big molecule. It contains very different types of structures, and it's a protein that can interact with many other proteins. Of course, we all know about factor VIII, but there are also other proteins like, for instance, angiopoietin-1, osteoprotoserine. There are some complement proteins that can bind to VWF. There is PSDL1 that can bind to VWF. So there are many proteins that VWF can interact with. So either directly or indirectly, it can be interacting with all kinds of physiological processes. But this is a figure that comes from a review that we wrote 10 years ago, so it's maybe already not very up to date anymore. But here you can already see that apart from its role in hemostasis, it can also function, as I say, as a molecular bus for various proteins. We know it has some pro-inflammatory activity. It can interact directly with leukocytes. It can function as an intermediate between platelets and leukocytes to attract leukocytes to sites of inflammation. We know it can influence angiogenesis. We know it can induce some apoptosis in tumor cells. And one of the things that we have been interested in the last several years is its interaction with smooth muscle cells. And that is what I will talk about in this presentation. So when we have VWF, most of us have usually focused on its role in plasma, its role in endothelial cells. But one of the compartments where we know little about is VWF in the subendothelial compartment. And when you look here at the figure here in the bottom, when you make a section of a vessel and you start to stain for VWF, you can see VWF in the endothelial cells, but you can also see that it's penetrating much deeper in the vessel wall. So what its function there, we don't know much about it. We know that it can help to attach the endothelial cells to the subendothelial matrix. We know that VWF can interact with some of the extracellular proteins that are also present in the matrix, but other than that, we don't know much about it. But one of the interesting things is that if we have a vessel that is being injured, one of the things that is happening very often is that smooth muscle cells start to proliferate. And at the same time, what we see, and this is some images from animal models, and it's mostly work in the 90s where that had been what the people were looking at, is that you can see that VWF is starting to penetrate into the subendothelial matrix. So at some point there is some interconnection between the smooth muscle cells and the VWF that is going deeper into the vessel wall. So that was something that we were interested in. So we started to look into that into more detail. So we analyzed first quite some human tissues from a biobank, from people with atherosclerosis, people who have gotten a stent, and things like that. And when you then start to stain for smooth muscle cells using smooth muscle cell actin, you can see that the cells that these smooth muscle cells that are starting to proliferate, the media is getting very thick. And at the same time, when you start to stain for VWF, you do not find your VWF only in endothelial cells anymore. But when you look here at the in the left image, you can see that VWF is penetrating very deeply, and you find it at the same sites where you also have proliferating smooth muscle cells. So this is one example, but we have many of these type of examples. So what we did to see if there was any connection between VWF and these proliferating smooth muscle cells, we did a similar experiment, but using our VWF deficient mice. We used two different models. So what I show here is the carotid ligation model, where you induce an injury in the carotid, but we also did an femoral artery denudation model and we got exactly the same results. So if you have a normal vessel where there is no ligation, you can see it looks very healthy with endothelial cells, and then here the smooth muscle cells around it. However, when you make the ligation, then you see that the vessel starts, that the opening becomes very smaller, and it's because these smooth muscle cells start to proliferate. The funny thing is, when you do that with the VWF deficient mice, there is actually no extra proliferation of the smooth muscle cells anymore. You can see here there is a lot of smooth muscle cells that are infiltrating into the intima, whereas here the intima stays very small. It's not extended at all, and you can see that having VWF allows smooth muscle cell proliferation, whereas in the VWF deficient mice there is no proliferation anymore. So does VWF have the capacity to induce smooth muscle cell proliferation? Well, that you can do very simply in in vitro. So you seed cells, you synchronize them for 6 hours, and then you add VWF, and in this case we also added a positive control, PDGF, and you look for 24 hours if there is any increased proliferation to the condition where you had nothing. So we call that the proliferative index. So without any addition, the cells proliferate, but very slowly. If you add an agonist, PDGF or VWF, the cells will start to proliferate. And indeed, when you add VWF on different concentrations, there is increased proliferation of VWF. And actually, the extent by which VWF can accelerate smooth muscle cell proliferation is as powerful as what we see for PDGF, which is a very strong inducer of smooth muscle cell proliferation. So the next question was, okay, if VWF is doing that, by what kind of mechanism can it stimulate smooth muscle cell proliferation? Well, one candidate that was very likely to be looked at was alpha V beta 3, which is an integrin that is expressed abundantly on smooth muscle cells. And we know that in the C4 domain of VWF, there is the RGD sequence that can interact with this type of integrins. So what we did, so first thing we did was we silenced alpha V beta 3 in smooth muscle cells and looked what happened with VWF-induced proliferation. And indeed, when you use scRNA for alpha V, then actually you block proliferation by VWF. So that was a good sign. We also used a specific peptide, the RGT myristylated peptide, which is a specific inhibitor of alpha V beta 3 signaling. So when we use that peptide, we could also block this proliferation induced by VWF. So at least we know that alpha V beta 3 is involved in this stimulating process by VWF. However, we started to run a bit into trouble when we were starting to block the interaction between VWF and alpha V beta 3. So we used two different approaches, an RGD peptide and an antibody that specifically blocks binding to RGD sequences. And actually, in both cases, although we knew that we could block binding of VWF to alpha V beta 3, it didn't disturb the proliferation at all. So that complicated a bit our lives. So what we knew at this point is that alpha V is involved, alpha V beta 3 is involved in a proliferation induced by VWF, but apparently there is another component, another receptor that is involved, and that is actually involved in the binding of VWF and then relays its signal to alpha V beta 3. So it's a two receptor process. So at that point we needed to look for a second receptor, and in a way we didn't need to look very far, because from our previous work we knew that LRP1 is actually involved in the clearance of VWF. So we know that VWF can bind to LDL-like receptors. And the problem is that VWF only binds to LRP when it's in its active conformation. And in our experiments, VWF is not in its active conformation. So we need to check which kind of LDL receptor would be involved in this process. So we looked at what in the literature what is known about LDL-like receptors on smooth muscle cells. So we chose several that are known to be expressed on smooth muscle cells, and so 3 of them, LRP1, LRP8, and LRP5, they actually do not interact with VWF under these conditions of non-shear stress. But there is actually one LRP4 that is expressed on smooth muscle cells and binds very efficiently to VWF. And indeed, when you look on these smooth muscle cells and you co-stain for LRP4 and VWF, you can see that there is a lot of yellow here, meaning that VWF and LRP4 indeed interact at the surface of smooth muscle cells. So we then looked if LRP4 is involved in proliferation of smooth muscle cells by VWF. So we used again an LRP4 scRNA, and again we could block this proliferation induced by VWF. So do LRP4 and alpha V beta 3 indeed localize at the smooth muscle cell surface, which is a requirement if one binds VWF and relays a signal to the other one to induce proliferation. So for that we used what we call a proximity ligand assay, which allows the detection of 2 molecules being together within a ratio of 40 nanometers. So what's happening, you have 2 antibodies against each receptor. They're recognized by 2 other antibodies that contain a oligonucleotide. And when they are close enough, those 2 oligonucleotides, they will hybridize. And via a PCR reaction, you can amplify these oligos using, and when you have these oligos being probed with a fluorescent marker, then you can see if those 2 are together. And I mean, it's no surprise that I show this, because you can see already in this image there is a lot of red dots on these smooth muscle cells, showing that alpha V beta 3 and LRP4 are indeed very closely together. So they are capable of relaying the system from one to the other.
What are the blue circles in this picture?
Well, actually, those cells are also stained with DAPI, which is a marker for the cell nucleus. Yeah. So those things are the nuclei of the cells. So we did quite some more work on what's happening in these cells during proliferation, or the signaling cascade that is involved. But I will try to summarize what is happening here on this cartoon. So what we now know is that actually VWF, that is in its global conformation, can actually bind to LRP4. And we now know that it's the A2 domain who is doing that. When it's binding to LRP4, LRP4 relays its signal to alpha V beta 3. And via alpha V beta 3, there is a whole signaling cascade being initiated involving SARC1, P38 MAP kinase, AKT, and ERK1/2. And actually that results in the proliferation of the smooth muscle cell, and that is resulting in the neo-intima that are being formed. So I think this is a very nice example how VWF has another function completely independent of primary hemostasis or factor VIII chaperone function. It's something very different, and it shows that VWF, when it's present in the subendothelial matrix, Well, it can start to interact with other types of cells. It can start to interact with other types of receptors. And I think this is only the first layer of a function of the subendothelial VWF that we have been able to uncover. And I'm sure that when we start to dig deeper, no pun intended, when we start deeper into the subendothelial VWF, we will uncover other functions as well.
I wonder if any patients with Willebrand disease has mutation in this binding sites, this LRP binding site or so.
Well, that is one of the things we want to start to go looking and to see if that has any effect on their capacity to develop atherosclerotic lesions or things like that. Mm-hmm. Yeah, but we have not. We know it's the A2 domain. So the next step, what we want to do, but that's not very easy to do, is to express mutants of the A2 domain and then try to see if they have a defective proliferation of the smooth muscle cells. Yeah. Okay, so now I want to go to the last part of my presentation about which is more focused on VWD. So VWD, it's a disorder that has a high prevalence, dependent on how you're looking at it, of course. When you just look very basically at VWF levels, if they are below the normal range, then you can quickly arrive at 1 per 100 when you look at the epidemiological studies. When you consider VWD only clinically relevant, when they have clinically relevant bleedings, then it's 100 times less prevalent, 1 per 10,000. And I think that's a very nice indication that shows that the prevalence of VWD is very difficult to estimate because it will depend on what criteria you will use. And it also shows that VWD is actually a very heterogeneous disease. It has a very heterogeneous character. So why is it so heterogeneous? Well, there are different ways where you can look at it. First, it is not always very easy to diagnose. The phenotypes are not always the same. So patients who may have VWD and who see their primary physician, they may not always be referred for further investigation to see if they have VWD or not. There's not always a consensus on the bleeding severity. For what one may think is a severe bleeding disorder or other things, well, you have maybe some minor bleeds, you just have to live with it. Not all the labs are equipped to diagnose VWD correctly. And also the genetic analysis is not always straightforward. The other thing is that VWF levels can be quite variable, even within one individual. It can depend on the time of blood sampling. It can depend on blood group and other genetic factors, stress, inflammation, hormones, exercise, they all influence VWF level. So especially those who are borderline VWD, or we can say, they can have VWF levels that are in the normal range one day and below the other day. So there is an incomplete recognition of patients with VWD, and that is particularly true for women. And those patients, they get suboptimal management. And of course, that will result in increased disease burden and a reduced quality of life. So why is VWD disproportional to women? Well, 70% of the symptomatic patients that are diagnosed with VWD are women. And why is that? Because especially the menorrhagia that is in these patients is very much developed. And in men, they don't have these sex-related bleedings. So that can be one of the reasons why we diagnose more women compared to men. The pregnancy that these women have and childbirth, they can be associated with severe bleeding complications. One reason why VWD is maybe still underrepresented in VWD, although we know there are more women already diagnosed, is that for some women with menstrual bleedings, they are very much underappreciated. That can be within the family. It's not always easy to talk about menstruation within families. And sometimes from generation to generation, it always has been like that. So sometimes girls are being said, well, but we always have been dealing with that, so you deal with it too. And I also know stories from clinicians who say, yeah, but you know, it's just a few days a month, deal with it. There is nothing to do on it. So that is a complication, and I think in the last several years, and especially Paula James and Dr. James and Dr. Ryan from Canada and the US, they are very much discussing these issues of women-related bleeding complications. So that's good that it's getting more attention, and let's hope that it will change the appreciation of these women. And why is it important? Because when you have these heavy menstrual bleedings, that is associated with a significant morbidity. There is the reduced quality of life, you miss school days, you miss work. It's known that there is more frequent hospitalization, there is an iron deficiency because of the bleedings, and there is an increased need for blood transfusion.
In December, we invited Paula here. She was with us in the webinar, and we discussed the importance of the genetic diagnosis and bleeding in women.
Yeah. Yeah, I mean,
it's a very important issue. I
know. I'm not a physician, so I don't see patients. But once in a while, here in our lab, once every year, we invite a group of patients. It's without clinicians; it's only researchers and patients. So we discuss with each other. We show them what we do here in the lab. In the afternoon, we listen to them. Actually, then we ask them about their life, how they live with their disease. And one of the things that from the first time on that we started to do that was that those patients with VWD, especially the women, they feel so underappreciated. They have so much troubles with their monthly menstruation, how they deal with that, with going to school, with going to work, with having boyfriends, etc., etc. And since then, I put this issue in all my presentations on VWD because it deserves more attention.
Yeah, I appreciate it very much. I believe in this as well. Like, if we consider the bleeding woman, probably the frequency of the von Willebrand disease will be higher in the world.
Yeah. So the question is, why don't we do anything about it?
Yeah.
I will try to come to that in the end of my presentation.
Yeah.
Yeah. So one of the complicated thing about VWD is its heterogeneity. So here I have summarized this kaleidoscope, as Dr. Suzanne, with whom we collaborate a lot, always says. We have type 1, 2A, 2B, 2M, 2N, and type 3. They all have different causes of different functional problems. In type 1, we have low VWF. In type 2A, we miss the high multimers, etc., etc. So, dependent on the genetic defect, one or another aspect of the VWF of protein is affected. And I will go quickly through, I don't know if it's necessary, but I will go quickly through the different subtypes that can be distinguished. So we have type 1 VWD, where the VWF is functionally more normal, the multimers are normal most of the time, but the levels are below the normal range, and also factor 8, apologies for the typo, is reduced. And what is important here is that we can distinguish more or less two types of type 1. The type 1 where VWF levels are below 30%. In that case, there is no doubt that it's really VWD, because in more than 90% of the cases there are mutations in the VWF molecule. However, there is a gray area where, unfortunately, where we find most of the patients, where the range is between 30 and 50%. Their mutations in VWF are less frequent, but they can be somewhere else in the genome that may be affecting VWF. This is mostly related as low VWF, and it is mild to moderate bleeding tendencies, and sometimes it's even referred as a risk factor rather than VWD. The causes for type 1 VWD are mostly reduced synthesis and secretion, or increased clearance. And in the last 15 years or so, it has become apparent that clearance is really an important cause for type 1. So we now officially know, this is now officially also known as VWD type 1C. Then we have type 2A, where we have loss of the high molecular weight multimers. In these patients, antigen levels may be quite variable, but often they are reduced, but you can find patients with normal antigen levels. But if the levels are reduced, then factor VIII is also reduced. We can distinguish two types. The type 1, or also 2E, is impaired dimerization of multimerization. And then group 2 is type 2A, where there is increased degradation by ADAMTS13. And usually in the type 2A, the loss of multimers is more important than in the group 1. From the work by Dr. Haberichter, we also know that in some patients who have impaired multimerization, they also have increased degradation. It's a combination of both. Then we have type 2B, where there are actually multiple defects, where there is a variable effect on antigen levels. There is fluctuating thrombocytopenia. In some patients, there is always thrombocytopenia. In other patients, there is only thrombocytopenia when they are pregnant, or where there is an inflammation, or where there is a shear stress, or a shear, or no, when there is stress in the patient. In some cases, especially the severe cases, there is reduced platelet function, and in also a number of patients, you can find giant platelets. So what is causing all these multiple defects? Well, there are mutations in the A1 domain, and what we call gain of function mutations. They enhance the interaction between VWF and platelets. They may have increased degradation by ADAMTS13. And what is very important, they also are characterized by impaired proplatelet formation, which contributes to the reduced platelet counts. And they also have impaired signaling, resulting from reduced activation of alpha-2b beta-3, also known as 2b3a. So they have everything together, I would say. They have those large platelet aggregates that you can find in the blood smears. The platelets don't work very well anymore. They have reduced antigen. The high multimers are gone. So what I understand from most clinicians is that the type 2B is the most difficult one to treat, because very often they would need platelet transfusion, VWF transfusion, in order to stop the bleeding. Am I right here?
Yeah, despite it is very easy to diagnose genetically.
Yeah.
It is easy, but it is difficult to treat, unfortunately.
Yeah. Then we have Willebrand disease type 2M. So the antigen in general is normal or maybe somewhat reduced. They have normal multimers. They are only characterized by reduced binding to functional ligands. In most cases, it's ligands, it's platelets, but it can also be collagen. Where is that coming from? Well, we have mutations in the A1 domain that impair binding to GP1B. We can have mutations in the A1 or A3 domain that impair binding to collagen. And that is something extremely rare. I think there are only 4 or 5 mutations known right now, where mutations in the C4 domain actually affect binding to GP2B3A. And then we have type 2N. Also here, antigen is in general normal or somewhat reduced. Multimers are normal, but they have a very strong reduced binding to factor VIII. So factor VIII is cleared more rapidly in the patients, and they have reduced levels of factor VIII. This is caused by mutations in the D'-D3 domain that impair binding to factor VIII. And there is type 2-3 where antigen is virtually absent and factor VIII levels are severely reduced. Well, that's why I have this white slide here. There is no VWF, so that's very good.
No expression at all.
Yeah. So so now we have talked about the burden of VWD in patients. They have reduced quality of life, especially women. So what can we do about it? Well, when we look at treatment options for VWD, what we have today is actually very limited. So we have Willebrand factor concentrates, which you need to infuse intravenously. They have a short half-life of less than 20 hours. They are expensive, and you really need to do intravenous application. You cannot give it otherwise. We have DDAVP, which you can give either intravenous or intranasal, or subcutaneously also. But its use is a bit limited, actually, because you cannot do repetitive administration. So you can either do it preventive when you know that there is an intervention to be done, or you can do it a bit on demand if there is a bleeding in the patient. But we know that there is a variable interpatient response. It doesn't work very well in the type 2A or the type 2M. In the type 2B, it's contraindicated, and it doesn't work in the type 3. So it's mostly used for the type 1 patients. Otherwise, we have antifibrinolytics, which is mostly used as an adjuvant therapy, and it can be efficient as a standalone for minor mucosal bleeds. So these are basically the things we are working with, but we're doing that already for 30 years. And if you look at hemophilia A, we have had a revolution of new treatment options. We have emicizumab, there is fitusiran coming, there is BIF01 with a very long half-life that is coming, there is gene therapy that is probably going to be approved this year or maybe next year. So for VWD, for the last 30 years, we are using VWF concentrates and DDAVP. There is nothing new. Yeah, we have recombinant, but it's doing the same thing as the old concentrates. So why are we always trying to think of we need to use the same molecules for all the different diseases? Should we not try to look at more specifically for the different subtypes? The groups will be smaller, but maybe we can be more efficient in treating a type 2A compared to a type 2M or a type 2B. So one of the things we have been thinking about for the last 5 or 6 years in our lab is what can we do to improve this type of treatments? And together with Cécile Denis and Sophie Sussen, we have written this review in Blood 2001, VWD, What Does the Future Hold? Where we were especially describing new types of treatments that you can think of. So we've been looking on, do I still have some time to talk? I'm not sure how much time I have left.
I think we have times until 2 o'clock.
Okay. Mm-hmm. So you can think of short-term type of treatments, which probably are mostly protein-based, but there is also now an aptamer that looks promising, where you specifically look for VWF or factor VIII. And in the long term, you can look at genetic approaches like gene therapy, gene editing, or transcriptional silencing. Well, I will not have the time to discuss all these different options and etc. So if you want to learn more about this, then I really refer you to this review, where each of these different approaches are being discussed in detail. But what I want to discuss today is actually something that can already be applied today, if you want, in a way, and that is emicizumab. So emicizumab has been developed for treatment of hemophilia A, especially severe hemophilia A. So why would you want to use it for VWD? Okay, so what is emicizumab? So emicizumab is when you normally look at factor VIII, what is the role of factor VIII in coagulation? Well, it's a cofactor for factor IXA, and it brings together factor IXA and factor X, and it's doing that a bit in a complicated way, where the activity of factor IXA is stimulated. But what does emicizumab? Well, it takes over at least one portion of what factor VIII can do, in that it can bring back together factor IXA and factor X at the phospholipid surface. And we know that in hemophilia A it works pretty well. It prevents a lot of the spontaneous bleeds that patients have. So how does that relate to VWD? Well, when you think of many of the VWD patients, they have reduced levels of VWF, but they also have reduced levels of factor VIII. And basically what we do not know is if patients with VWD that has low factor VIII and low VWF, when they start to bleed, what is actually the contribution of a lack of VWF? And what is the contribution of a lack of factor VIII that contributes to this bleeding in the patient? So can you think of when we start to improve at least factor VIII in these patients, can that increase the hemostatic potential of these patients already? Well, does that make sense? Well, for that, you can actually look in some case reports that have been presented in the literature, where patients who had Willebrand disease type 3 and also had, they are not lucky, also had antibodies against VWF, they needed surgery. So you cannot give those patients VWF concentrates. So what did the clinicians do? Okay, but at least try to normalize factor VIII in these patients. So they were giving a lot of factor VIII during the surgery. And in each of these cases, the patients had very little bleeding, surprisingly. So that means that in these patients under these conditions, the factor VIII deficiency appeared to be more important than the VWF deficiency in a way. And
but as far as I know, the factor VIII and von Willebrand factor serves in different parts of the hemostasis. Like for the primary hemostasis, we need to have functional von Willebrand. For the coagulation, we need to have functional factor VIII.
Yeah, completely correct. Yeah.
But like giving higher amounts or treating the von Willebrand patients with factor VIII, how will maintain the primary hemostasis?
Yeah, that remains still an issue. But what you're looking at in these patients in the end is how much blood do they lose during the surgery? Is it manageable, or do they bleed to death in a way? Yeah. And in these 4 patients, having the factor VIII corrected, that reduced the blood loss considerably and didn't need any additional VWF. No? Mm-hmm. I'm not saying here that you should give patients factor VIII and they will not bleed the rest of their life. Mm-hmm. That's not what I'm saying.
Yeah,
I'm saying here is under these conditions, correcting the factor VIII very much reduce the blood loss. So that means that the blood loss in these patients during this type of
I have a question. This maybe like similar to von Willebrand factor. Factor VIII has additional roles that we don't know.
Yeah, exactly. So, if factor VIII is correcting the hemostatic potential in these patients, you can think, hey, why are we not giving those patients just factor VIII as a prophylaxis? But of course that will not work, because the half-life of factor VIII is way too short to think of it as a prophylactic treatment approach. Because the half-life is only 2 hours when you have no VWF. And there is where amicizumab comes back in the picture, because amicizumab has a very long half-life of about 4 weeks. It can be given subcutaneously. And so even if we have a deficiency of VWF, can we then at least not try to reduce the burden of bleeding by correcting the factor VIII deficiency by using amicizumab? Well, there are now a few case reports where they have tried to do that. And this is the first example that was published by Dr. Wyant in 2019. It concerned a 5-year-old boy who was first given VWF factor VIII concentrates 3 times a week. And unfortunately, the boy developed inhibitors against both VWF and factor VIII. So that was an issue, and he started to develop recurrent ankle bleeds. So he had to be immobile for most of the time. So they tried to use some off-label treatment using recombinant 7A. That didn't resolve the ankle bleeds. Then they shifted by using FIBA, APCC, using a portacat. That was working, and he had less bleeds, but the daily infusions were actually giving a very high treatment burden, affecting the quality of life of the young patient, his parents, etc. And then they decided, okay, but if we now have amicizumab, can we not try amicizumab in the boy? So they gave him amicizumab standard conditions, 3 milligram loading dose for 4 weeks, and then 1.5 milligrams per week further on. And actually, the boy did not have spontaneous bleeds anymore for a period of 9 months. So all the ankle bleeds, they were resolved by using amicizumab. Well, that was the first example. There are now more examples available. Some patients were young, some patients were older, 48 years, but most of them were children. There were alloantibodies in 4 of the patients, but not in 2 other ones. Most of them were characterized by joint bleeds and joint swelling. Some had also mucocutaneous bleeds. And actually, upon starting treatment with amicizumab, without exception, they showed hemostatic improvement. Follow-up is relatively short, between 1 and 12 months. And the patients that I'm aware of, they did not have any joint bleeds anymore. Some needed some adjuvant treatment because they had a tooth extraction or things like that. Some had a mild mucocutaneous bleed that needed to be treated. But all the joint bleeds, they are resolved by using amicizumab in these patients. So when I presented this last 2 weeks ago at the World Hemophilia Federation Congress, there was also a clinician came to see me after my presentation. He said, well, I have a VWD type 3 patient on amicizumab now for 3 years. She only needs, does not have antibodies against VWF or factor VIII, but she's on amicizumab that works perfectly. From time to time, when she gets a mucocutaneous bleeds that is a bit more severe, we give her VWF, but most of the time she doesn't need anything else. Before she had a lot of joint bleeds and a lot of mucocutaneous bleeds. So there is really, I think, an opening for an existing treatment that can be applied to VWD. It seems to be a viable option to reduce most of the bleeding tendency in type 3, as is apparent from these case reports. And actually in our lab, we have been treating our VWD type 3 mice with amicizumab, and we see a strong reduction in the bleeding tendency in these mice. So there is really something going on with amicizumab and type 3. But of course, these are anecdotal presentations, and I think it would be very wise if some kind of in-depth clinical trial is being set up to test the use of amicizumab in Willebrand disease type 3. I have to say, I was skeptical in the beginning too, but seeing what we see in the type 3 mice and seeing what and hearing from the clinicians what they see with amicizumab, I think it's a no-brainer that this should be done. It will not resolve all the bleedings in these patients, but at least some of them. And especially the joints will be preserved much better. But always keep in mind that it will correct the factor VIII deficiency and not the VWF deficiency. Does it work in other types of Willebrand disease? That we do not know. I think it will need to be tested. Type 2A, 2B, 2N, 2N. I think 2N is a no-brainer as well. For the other ones, I don't know. And it will require additional studies.
Yeah, 2N is also associated with the factor VIII deficiency.
Yeah,
therefore, it should work in these patients as well.
Yeah. Type 2A, 2B, 2M, I don't know. That remains to be seen, because there the factor VIII levels are usually pretty okay already. So adding amicizumab, I don't think it will help a lot. No? So I think this was my last slide. Yeah, thank you for your attention. And if you have any additional questions, I would be happy to reply to them.
Hey, doctor. Yes, hocam, I have a question.
Yeah.
Yes.
Can you turn off your microphone, and I will let you ask your question. Thank you very much for your talk. It was really informative for me. I learned new subjects, including the new role of Willebrand factor. And I have a student, Nazlı. She would like to ask a question to you.
Yeah.
Nazlı, please go ahead.
First, thank you, doctor. It was a pleasure to us to listening you. And I know that VHA is a complex disease with a clinical and genetic heterogeneity. And I am wondering that is VF factor treatment 100% effective using gene therapy?
Well, gene therapy for VWD, it's a very good idea, but it's very far away. And I will try to explain you why. So for factor VIII, you know, it's a large molecule, and we already had to take out the B domain for it to fit into the AAV factors that are being used for gene therapy. The problem is that the most used virus for gene therapy, the AAV, cannot contain a genetic probe that is more than 5 kilobase long. By taking out the B domain of factor VIII, it gets to a bit 4.5 kb, and then you need some other things. So that just fits into an AAV. The mRNA of VWF is 9 kb. And there we do not have a B domain that we can take out. So it doesn't fit into the AAV. So one of the things that we have tried is actually to cut the cDNA in 2 parts, put some recombination probe on each side, and then used what we call a dual factor approach. So we tested that in our mice, and indeed you can see VWF being expressed in these mice, but we do not get higher than 1 or 2%. So that's not enough. It's not a clinical viable approach for yet. So we are thinking of other strategies to make it better. But that's too early. But if we could do that, that would be wonderful. But even if we would be able to do that, we still have another problem to solve. Because if you want to express your VWF in the endothelial cells, because we know already when we do that in the hepatocytes, it doesn't get multimerized enough. So you need to really go to the endothelial cells to get the high molecular weights. The problem is for type 3 already, at least in half of the patients, they express what we call a dominant negative VWF molecule. So that is a molecule that impairs the multimerization of your healthy molecule, your healthy subunit. So even if you start to express more healthy VWF, the presence of the dominant negative variant will impair your multimerization anyway. So that's also a problem we need to conquer. So it's complicated. Summarized.
Thank you, Nazlı. It was a really good question. And personally, I don't know. For me, the gene therapy is like people are expecting too much from gene therapy. But it is too expensive, first of all, and it is not applicable in all over the world, especially for the developing countries. It is really hard to reach.
Yeah, no, I agree. Even from the patients that I know, some are completely, if it's available, they really would like to go. Most of them I hear, I think I wait for a third or fourth generation, which they are.
And this is what I was going to say. At the first sight, it is expensive, and in the second sight, we don't know the long-term effects. And we follow the patients for hemophilia B, for example, just a few years. Like I don't think that it has been 10 years.
Yeah,
and I heard that the amount is getting low.
For factor VIII, it's going down. Yeah, so we don't know when it stops going down, or if it's going to stop going down.
Needs to be repeated in 10 years or 20 years. Then it is not a gene therapy for me.
Yeah. No, that's true. And yeah.
Nazlı, do you have any other questions or comments?
Actually, hocam, I had,
yes,
but
please.
You enlightened me, and I'm wondering that the gene therapy is an interesting method to use any disease, and I'm thinking that we can find a treatment to all diseases, and for example, CRISPR is a amazing method, and if we use that method in gene therapy for von Willebrand factor disease, it would be great. But I will search that. It's incredibly amazing. It's looking like that to me. And thank you for your presentation, Doctor. I'm very, very thankful for you.
She's right.
Welcome. I want to follow up on your comments. I mean, CRISPR, it's a great tool, and I'm sure it's going to change the way we look at all the different diseases. Actually, in our lab, we try to set up a CRISPR-Cas9 approach for some of the VWD types. Would you be able to use it combine it with a gene therapy approach where you can use gene therapy to change the genome of the patients? I think maybe in 10, 20 years, I would guess that would be possible. There are a number of technical hurdles that need to be taken. You don't want to have off-target effects. You need to target the cells specifically. You don't want to happen it in other cells than the cells you want, etc., etc. When I come back to the gene therapy, so I said, gene therapy for VWD as a whole, that's difficult because it's so big and all kinds of troubles. But that is where the change of mind should occur. Should we not target the subtype specific things? If we think of type 2N, one of the easiest things, one thing that we can do today, we can start doing that today. If industry wants, we can do it today. we can start to express the prototype with the D prime D3. It's very small, it fits in an AAV, you get very high levels of expression, and you will normalize factor VIII levels in these patients. So they are not type 2N anymore, they are normal. So that we can do that now. For type 1, actually we have developed now a molecule, which I presented last week, or 2 weeks ago at the Congress. We've made a bispecific nanobody that can bind to VWF and albumin at the same time. If we give that to a mouse with the type 1, so who has 12% VWF, we double with one subcutaneous injection, we double VWF levels for a period of 10 days. 10 days, it stays up. Yeah, 10 days. 10 days, it stays up. We can put this bispecific nanobody, which is a 30 kilodalton protein, we can put it in an AAV and give that to the patient. And all the patients with type 1, they will be normalized. So, but the point is, we need to change the mind of the people who are involved in developing gene therapy. I mean, I can do it here in my lab. We do it already. I can cure VWD. The problem is we need the help of clinicians. We need to have the support of the patient organizations, and industry needs to do it. I cannot do it by myself.
You are right. Yeah, like when we say gene therapy, people, I think that it is the therapy including all gene, but we can use the part of the gene to treat the diseases.
Yeah.
Yeah, you should change the perspective maybe, and especially in pharmaceutical companies.
Yeah. Or we should do it ourselves.
Yeah.
Let's try.
As a science or scientific tool, I think CRISPR-R and other techniques, the vector-derived vector delivery, are really very interesting, amazing techniques. But we should also think of the other benefits that we can get from gene therapy or other types of therapies that you mentioned.
Yeah.
And any other questions from the audience? Maybe I misunderstood from your presentation, like one of the functional Willebrand factor is the negative regulation of angiogenesis. Is it correct?
Yeah.
But another novel function of Willebrand factor is to promote proliferation of smooth muscle cells. Isn't these two are contradictory, or I am missing something?
Well, I think I think it's two separate processes. So, okay. So I don't think we should try to mix them up. Mm-hmm. But actually, the angiogenesis part is much more complicated than we thought.
Okay.
Because if you look at the different animal models, mm-hmm. dependent on the model you use and dependent on where in the animal you are looking at, VWF can be pro or anti-angiogenic. So it can actually have opposite roles in the same physiological process. And what we do not know is why it's doing pro-angiogenic on one time and anti-angiogenic the other time. So that's why I didn't go into the angiogenesis, because it's not my specialty, and I'm confused by it myself already. So I didn't want to confuse you more than necessary.
All right, thank you. And I think I asked all the questions. Other people should ask questions. Nazlı, do you have any other questions or other questions from the audience?
Hocam, actually, I have one more.
Okay.
Dr. Peter said there was a drug, but I forget its name now, and it's ending with UMAP, and I have already seen this type of drugs with ending UMAP, daratumumab, for example.
Yeah. Okay.
Yes.
So,
Derived from monoclonal antibodies.
Yeah. So UMAP means that it's a monoclonal antibody. And actually, you can learn a lot from the way the names of the antibodies is described. If you have C, a C with an I, then it means that it's against a circulating protein. Yeah. If you have UMAP, then it means that it's a fully human antibody. If you have ZUMAP, then it means that it's a humanized monoclonal antibody. If you have XIMAP, then it means that it has that it's a chimeric antibody where the variable portion is fully mouse and only the FC portion is human. So there is actually a rule to name your antibodies dependent on how your antibody has been developed. The only portion that you are free of is the first, before you start to XISUMAP or CISUMAP or CIMAP or SUMAP. Yeah, there are some papers in the literature where you can learn that.
Actually, they have a take-home exam.
Okay.
About these monoclonal antibodies. So
I should have a slide somewhere where I will try to find quickly the slide where I have that.
Yeah, sure.
Written. Give me 2 minutes, huh?
Yeah, sure. Go ahead.
Where is it? Where I don't find it. Yeah, I saw it. Yeah, I have it. I'm opening it. It's a big presentation, so it takes a bit of time. It's on its way. Where does it stay? If there are in the meantime some other questions, then
yeah. Any other questions from the audience?
It's opening. Yeah, I have it. Okay, I will share. Mm-hmm. So here is the slide. So this is the nomenclature of antibodies. If you have UMAP, then it's a fully human antibody. If you have ZUMAP, then the whole antibody is human except for the variable regions, the CDRs, I need to say. If you have XIMAP, for instance, Abciximab, which is an anti-2B3A antibody, ReoPro, which I think is the other name, then everything is human, but they have not changed the variable domains here. And if you have a fully human, fully murine antibody, then it's an OMAP. And that was the very first one, OKT11 in the past, which was that one. And the only thing you need to put before ZUMAP or UMAP or XIMAP, if it's against a circulating protein or circulating cell, then you need to put C in front of it, from circulating.
Yeah, thank you for this information. I think these monoclonal antibody mediated treatments are better for me. This is my idea, in my opinion, sorry. They are better because if it is humanized or human-derived monoclonal antibody, then the immunogenicity is very low and there is no risk of inhibitor development, maybe.
Yeah. No, I agree. I mean, that's why we were started to focus to work on nanobodies, because they are very small. They're only 1/10 of the size of a monoclonal antibody. I mean, there is a caplacizumab, circulating humanized, which is used for TTP, and they can be given subcutaneously very easily. They go easily into the circulation, have a long half-life if you treat them correctly. And so, okay. Well, if there are no other questions, then
I think.
Can I ask one more?
Of course.
Thank you. And I'm wondering that do you express the drug in your laboratory or you buy it from other laboratories?
How do you mean that?
I think she is trying to ask you if you are manufacturing the monoclonal antibodies in your lab, or you are.
Yeah, that depends.
Commercially available, but how do you get them?
That depends. We have antibodies that we produce ourselves, but emicizumab, those I get from the clinic, I don't produce them myself. That's too complicated. But all the nanobodies and all that we produce in our lab without any problem. Yeah.
Okay, thank you.
Dear Peter, I think there's no other questions, and it has been almost 2 hours. I thank you very much. I wish we had these meetings face to face in here or in Paris.
Maybe we will see each other in London for the ISTH.
Yeah, I applied for it, but I don't think that I will be able to attend. One of my colleagues will present our work there. Probably I won't be there, but if I will be there, I will definitely catch you up.
Okay, that's good.
All right, so thanks again for your time and for this wonderful presentation, and maybe in the next year we will meet again on webinar or in other things.
Okay, that's good. Thank you very much again for the invitation, and I hope the people have enjoyed the presentation. And if there are any other questions, just send me an email.
Yeah, sure. Yeah.
Bye-bye.
Take care.